Monday morning. My trial has 45 active sites across the US, Germany, France, and Japan. 14 patients enrolled last week. 47 are currently in screening. 3 sites are on a recruitment hold. Meyer Osei, Arventa Global, Clinical Trial Manager. I run Clarity 301, our pivotal phase 3 trial for velaratinib in RET fusion-positive non-small cell lung cancer. Second line, randomized 2:1 against docetaxel. Primary endpoint, progression-free survival. 312 patients to enroll, we are at 184. My IND has been in effect for 3 years. Phase 1 established the dose. Phase 2 gave us the signal, 68% response rate in the RET fusion population. Now Phase 3 is the question FDA needs answered before we can file. Does this survival benefit hold in a randomized, controlled setting? This week, a monitoring visit report from our CRO at MD Anderson flagged 2 protocol deviations. A DSMB meeting is scheduled Thursday, safety data from the first 120 patients. And I have a protocol amendment in regulatory review that changes one of our secondary endpoints. Every day something changes. The protocol doesn't move. The data doesn't lie. The sites need support. Agentforce clinical operations tracking gives me real-time enrollment, deviation, and site performance data across all 45 sites. The clinical trial manager role, operational hub of a phase 3 trial. The clinical trial manager, CTM, is the sponsor-side operational lead for a clinical trial. In a phase 3 pivotal study, the CTM is responsible for site management, overseeing the network of investigator sites, managing site performance against enrollment and quality metrics, CRO oversight, managing the contract research organization that conducts day-to-day monitoring and data collection, regulatory document management, IND maintenance, protocol amendments, IRB correspondence, safety event tracking, ensuring adverse events are collected, assessed, and reported within regulatory timelines, and cross-functional coordination, clinical operations, regulatory affairs, data management, biostatistics, medical affairs, and commercial all intersect at the CTM. The CTM does not run the science, that is the medical director and clinical scientists. But the CTM ensures that the science is executed according to the protocol, on time, and in compliance with GCP regulations. Investigational New Drug Application, the legal framework for human trials. Before a pharmaceutical company can test an investigational drug in human subjects in the United States, it must file an investigational new drug application with the FDA. The IND describes the drug, chemistry, manufacturing, and controls, CMC, the preclinical data supporting human testing, pharmacology and toxicology, and the proposed clinical protocol, study design, patient population, endpoints, safety monitoring plan. FDA has 30 days to review the IND and place a clinical hold if there are safety concerns. If no hold is issued, the trial may proceed. An active IND is a living regulatory document. Every protocol amendment, every new investigator site added, every safety report, and every annual progress report, IND annual report, is submitted as an IND amendment. The CTM is typically the operational owner of the IND, ensuring that all required submissions are made on time and that the IND accurately reflects the current state of the trial. Phase 3 design, the pivotal randomized controlled trial. A Phase 3 pivotal trial is designed to provide the primary evidence of efficacy and safety that supports regulatory approval. In oncology, Phase 3 trials are typically randomized, controlled trials comparing the investigational drug against the current standard of care, in Clarity 301. Velaratinib versus docetaxel in the second-line RET fusion-positive NSCLC population. Randomization, here too, 2 patients to velaratinib for every 1 to docetaxel, ensures that patient characteristics are balanced between arms, allowing the treatment effect to be attributed to the drug rather than to patient selection. The primary endpoint, progression-free survival, PFS. The time from randomization until disease progression or death is selected based on prior FDA precedent for the indication, clinical meaningfulness, and feasibility within the trial timeline. The sample size, 312 patients, is calculated to provide sufficient statistical power, typically 80–90%, to detect a clinically meaningful difference in the primary endpoint between arms. The CRO relationship, sponsor oversight of outsourced operations. Most Phase 3 trials are operationally executed by a contract research organization, CRO, a specialized company hired by the sponsor, Arventa, to perform trial monitoring, data collection, and site management functions. The sponsor retains regulatory responsibility for the trial. The CRO executes under a delegation of authority. For the CTM, the CRO relationship requires active oversight, reviewing monitoring visit reports, tracking site performance metrics, managing CRO escalations, and ensuring that the CRO's work meets the sponsor's quality standards. FDA Guidance 21 CFR 312. 52 makes clear that a sponsor cannot transfer regulatory responsibility to a CRO. If the CRO makes an error, the sponsor is accountable. The CTM's oversight role is not passive approval of CRO deliverables, but active quality assurance throughout the trial. DSMB, the independent safety watchdog. The Data Safety Monitoring Board, DSMB, also called the Data Monitoring Committee, DMC, is an independent group of clinical experts, statisticians, and bioethicists that reviews accumulating safety and efficacy data in a blinded or unblinded fashion at pre-specified intervals throughout the trial. The DSMB has authority to recommend stopping the trial early. For safety, unacceptable harm in the treatment arm. For efficacy, overwhelming benefit that makes continuation unethical for the control arm. Or for futility, the trial is unlikely to achieve its primary endpoint even if completed. The CTM does not sit on the DSMB, does not see unblinded data, and does not receive DSMB deliberations, only the DSMB recommendations. Managing the DSMB relationship, preparing safety data packages, coordinating meeting logistics, implementing DSMB recommendations is one of the CTM's most critical ongoing responsibilities. The protocol is the law of the trial. Every decision made before the first patient is enrolled defines every decision that can be made afterward. Meyer Osei, Arventa Global, Clinical Trial Manager. Clarity 301 took 18 months to design. The core questions: what is the right comparator? What is the right primary endpoint? Who is the right patient population? Control arm, docetaxel. Standard of care in second-line NSCLC for patients who have progressed on first-line therapy. We considered pemiarixed and docetaxel combination, but FDA guidance for this indication precedents single-agent docetaxel. The control arm must be what patients would actually receive if they weren't in the trial. Primary endpoint, progression-free survival, not overall survival, because RET inhibitors in the first-line setting may affect what patients receive at progression, confounding OS. FDA agreed. PFS at a median of 4 months in the docetaxel arm, median PFS of 9 months in veliratinib in Phase 2, that's the signal we're trying to confirm. Eligibility criteria. RET fusion confirmed by local or central next-generation sequencing. ECOG performance status 0 or 1. Prior platinum-based chemotherapy. No prior RET inhibitor treatment. Each criterion is a clinical and operational decision. Too restrictive and you can't enroll. Too permissive and your population is heterogeneous. The protocol is a negotiated document. Clinical science, regulatory strategy, and operational feasibility at the same table. Agentforce protocol management links eligibility criteria to site screening data across all 45 sites. The clinical trial protocol, structure, and regulatory function. The protocol is the master document that governs every aspect of a clinical trial. A Phase 3 protocol contains the study rationale and background, study objectives and endpoints, primary, secondary, exploratory, study design, randomization, blinding, treatment arms, eligibility criteria, inclusion and exclusion, study procedures and visit schedule, safety monitoring plan, adverse event definitions, reporting requirements, dose modification rules, statistical analysis plan summary, and regulatory compliance statements. The protocol is submitted to FDA as part of the IND, to each institutional review board for ethics approval, and to regulatory agencies in each country where the trial is conducted. Any substantive change to the protocol, a change to eligibility criteria, endpoints, statistical analysis methods, or safety monitoring procedures requires a protocol amendment, which must be submitted to FDA, re-reviewed by IRBs, and communicated to all active sites before implementation. The CTM is the operational owner of the protocol, ensuring that all amendments are submitted, approved, and implemented consistently across all sites. Primary endpoint selection, the regulatory negotiation. The primary endpoint is the single pre-specified outcome measure that will be used to assess whether the drug is effective for regulatory approval. In oncology, the choice between progression-free survival, PFS, and overall survival, OS, reflects a balance of scientific, regulatory, and operational considerations. Overall survival, time from randomization to death from any cause, is the most definitive evidence of clinical benefit but requires longer follow-up, larger sample sizes, and may be confounded by subsequent therapies that patients receive after progression. Progression-free survival, time from randomization to radiographic disease progression or death, can be assessed earlier, requires fewer patients, and is accepted by FDA as a primary endpoint for accelerated approval or regular approval in many oncology settings, provided the PFS benefit is large enough to be clinically meaningful. The selection of PFS as the primary endpoint for Clarity 301 reflects FDA precedent in the RET fusion-positive NSCLC space and the magnitude of the Phase 2 signal, a more than doubling of median PFS, that suggests PFS benefit will translate to meaningful clinical benefit. Eligibility criteria, the clinical and operational tension. Eligibility criteria define who can and cannot enroll in the trial and are among the most consequential design decisions a protocol team makes. Inclusion criteria specify the characteristics patients must have: RET fusion confirmed by NGS, ECOG performance status 0-1, prior platinum-based chemotherapy. Exclusion criteria specify characteristics that would prevent enrollment: prior RET inhibitor therapy, active CNS metastases requiring treatment, inadequate organ function. From a scientific perspective, narrow eligibility criteria produce a homogeneous study population in which the treatment effect is more precisely estimated. But the population may not represent the patients who will ultimately use the drug if approved. From an operational perspective, narrow criteria reduce the pool of eligible patients, increase screen failure rates, and extend the enrollment timeline. FDA has in recent years encouraged broader eligibility criteria in oncology trials to improve trial diversity and generalizability. A design consideration that Clarity 301 incorporated by allowing a broad range of RET fusion partner genes and accepting both local and central NGS confirmation of the molecular alteration. The control arm, the standard of care requirement. The comparator arm in a randomized controlled trial must represent what patients would receive in standard clinical practice outside the trial. The ethical basis for randomization is that neither arm is known to be superior at the time of enrollment. For a second-line NSCLC trial in patients with RET fusion-positive disease who have progressed on platinum-based chemotherapy, the standard of care at the time Clarity 301 was designed was single-agent docetaxel, an established cytotoxic chemotherapy with documented activity in second-line NSCLC. Choosing docetaxel as the comparator reflects FDA regulatory precedent in this line of therapy and the clinical reality of what patients would receive outside the trial. Control arm selection has direct implications for the statistical design. The assumed median PFS in the docetaxel arm, 4 months, based on published data, determines the hazard ratio that the trial is powered to detect and therefore the sample size required. Adaptive design elements, pre-specified flexibility within the protocol. Clarity 301 includes one adaptive design element, an interim efficacy analysis at 60% of the target number of PFS events, with pre-specified stopping criteria for overwhelming efficacy. If the interim analysis shows a hazard ratio below a pre-specified boundary, favoring veleratinib, the DSMB may recommend stopping the trial early for efficacy, allowing Arventa to file the NDA earlier and patients in the control arm to access the drug. Adaptive designs require pre-specification in the protocol and statistical analysis plan, the stopping boundaries, the alpha spending function, how the statistical significance threshold is allocated across interim and final analyses, and the DSMB process are all pre-specified and approved by FDA before the trial begins. An adaptive element that is added or modified after the trial is underway is not a legitimate adaptation. It is a protocol amendment that requires re-review and raises questions about data integrity. Before we enrolled the first patient in Clarity 301, we sat across the table from FDA. We needed to know, is this protocol approvable? Meyer Osei, Arventa Global, Clinical Trial Manager. 2 meetings. The end of Phase 2 meeting, Type B, where we presented our Phase 2 data and our proposed Phase 3 design. FDA reviewed our primary endpoint, our sample size, our control arm. They had questions about the RET fusion diagnostic strategy. Can a local NGS test qualify patients, or does FDA require central confirmation? We proposed a dual qualification approach: local NGS for enrollment, central confirmation for the primary analysis population. FDA accepted that. Then we requested a special protocol assessment, SPA. We submitted the Phase 3 protocol and asked FDA a specific question. If Clarity 301 is conducted as described and the primary endpoint is met, will this be sufficient to support NDA approval? An SPA is a binding agreement. FDA commits in writing to the protocol design. If we execute the trial as written and meet the primary endpoint, we have regulatory certainty, barring a major safety signal or public health issue. We got the SPA. That's the foundation Clarity 301 is built on. The SPA doesn't guarantee approval. It guarantees that FDA won't change the goalposts. Agentforce Regulatory Affairs Integration tracks SPA commitments, IND correspondence, and FDA meeting action items across the clinical team. FDA meeting types: Type A, B, and C. FDA classifies meetings with drug sponsors into 3 types based on urgency and complexity. Type A meetings are reserved for urgent matters, clinical holds, special protocols for life-threatening conditions, and disputes about FDA decisions, and must be scheduled within 30 days. Type B meetings cover the most clinically significant milestones in drug development: the pre-IND meeting, before filing an IND, the end of Phase 1 meeting, before Phase 2, the end of Phase 2 meeting, before Phase 3, and the pre-NDA/BLA meeting, before filing the marketing application. Type B meetings must be scheduled within 70 days of FDA receiving the meeting request. Type C meetings cover any other matter and must be scheduled within 90 days. The end of Phase 2 Type B meeting is arguably the most important meeting in a drug development program. It is the moment where the sponsor and FDA align on Phase 3 design before the sponsor commits hundreds of millions of dollars to the pivotal program. End of Phase 2 meeting, aligning on Phase 3 design. The end of Phase 2 EOP2 meeting is a formal meeting between the sponsor and FDA's review division, held after Phase 2 results are available and before the Phase 3 program begins. The sponsor submits a meeting package approximately 4 weeks before the meeting, containing a summary of the Phase 2 results, the proposed Phase 3 protocol design, the statistical analysis plan, the proposed patient population, the control arm selection rationale, and specific questions for FDA. FDA's written responses to the meeting questions, provided in the official meeting minutes within 30 days of the meeting, represent the agency's current thinking and provide regulatory guidance that the sponsor relies on in designing the Phase 3 program. The EOP2 meeting is not a guarantee. FDA can and does change its positions as new scientific evidence emerges, but it establishes a documented regulatory foundation for the Phase 3 program. For Clarity 301, the EOP2 meeting resolved the central diagnostic question: dual local/central NGS qualification, the control arm, docetaxel, and the primary endpoint, PFS, 3 of the most consequential design decisions in the protocol: special protocol assessment, the binding FDA commitment. The special protocol assessment, SPA, is a formal FDA process in which the agency evaluates the design of a specific clinical trial and commits in writing that, if the study is conducted as described and the primary endpoint is met, the results will be considered adequate to support regulatory approval, subject to the overall benefit-risk assessment. An SBA is a binding agreement. It cannot be unilaterally changed by FDA except in specific circumstances. Safety issue that wasn't apparent at the time of the SBA, a public health issue, or the sponsor materially deviates from the agreed protocol. For sponsors, the SBA is the highest level of regulatory certainty available before Phase 3. It eliminates the risk that FDA will challenge the trial design at the NDA stage after the data are available. SBA requests are submitted to FDA along with the complete Phase 3 protocol, and FDA has 45 days to respond. Not all FDA divisions offer SBA agreements, and FDA may agree with some elements of the protocol, but not others, issuing a partial SBA. The diagnostic strategy, companion diagnostic vs. local testing. Precision oncology trials targeting molecularly defined patient populations face a central operational challenge. How do you confirm that a patient has the molecular alteration the drug targets quickly enough to allow enrollment, reliably enough that the primary analysis population is well defined? In Clarity 301, the target population is RET fusion-positive NSCLC, patients whose tumors carry a chromosomal rearrangement that produces an oncogenic RET fusion protein. RET fusions can be detected by next-generation sequencing, NGS, fluorescence in situ hybridization FISH, or RNA sequencing. The dual qualification strategy in Clarity 301, local NGS for enrollment eligibility, retrospective central confirmation for the primary analysis population, allows fast enrollment. Sites don't wait for a central lab result, while ensuring that the primary efficacy analysis is based on centrally confirmed RET fusion status. FDA's agreement to this approach at the EOP2 meeting was a critical design decision. It determined the size of the confirmable primary analysis population and the statistical strategy for handling patients who enroll on local testing, but are not centrally confirmed. Regulatory strategy as operational input. The SBA and EOP2 outcomes directly shape every operational aspect of the trial. The CTM translates regulatory commitments into operational requirements. The diagnostic strategy agreed with FDA determines how sites collect and ship tumor samples and how the central laboratory is contracted and qualified. The control arm selection determines the docetaxel supply chain. The primary endpoint, PFS, determines the imaging schedule, CT scans every 8 weeks, and the independent radiological review process. And the sample size, 312 patients, determines the number of sites needed and the recruitment timeline. A regulatory agreement that seems abstract in a meeting room becomes a concrete operational requirement in the trial. The CTM's job is to understand the regulatory strategy well enough to identify where protocol commitments create operational risks, enrollment feasibility, site capability, diagnostic turnaround time, and to surface those risks to the clinical program team before they become trial-threatening problems.